CRISPR is a term. It's bacteria have an immune system that cuts invaders, cuts their DNA. And what we've done now, as scientists, we've now utilized that system, take it out of the bacteria and use it to create designer mutations, designer gene changes in animals and also in humans. So it's a bacterial immune system that corrects genes. And we use it all the time now. It's actually, what's interesting about it is we've been able to mutate genes for many years, but this is dial up a gene mutation. You can choose exactly where you want to make it. And recently, a Chinese researcher in our field came out and said he's engineered a couple of twin girls with CRISPR to be resistant to HIV, the AIDS virus.
The realm of DNA editing and medical genetics is evolving at an astonishing pace, surpassing our expectations. Artificial intelligence and CRISPR technology have been revolutionizing the field of medicine. AI is actively employed in identifying harmful genes and treating diseases, offering tailored solutions for specific cases and applications. The significance of AI in healthcare and its profound impact on the biotech industry are poised to grow exponentially in the near future. Multiple emerging gene editing technologies, alongside AI, are poised to bring about significant transformations in healthcare. Among these technologies is CRISPR, an abbreviation for clustered regularly interspaced short palindromic repeats. CRISPR empowers scientists with an unprecedented level of power and precision to modify human DNA, essentially reprogramming life itself. This newfound capability even extends to enhancing human intelligence. Before CRISPR was utilized on human embryos, scientists experimented with this technology on simpler animals. The aim was to validate its effectiveness. These experiments demonstrated that CRISPR could accomplish much more than previously envisioned. Scientists can employ CRISPR to alter, remove, or add DNA to any organism, spanning from human embryos to pigs to wheat crops. The mechanism involves targeting a specific part of the genome and introducing new DNA into it.
So CRISPR is an acronym for basically a system that is from bacteria that they use to kill and destroy the DNA of invading organisms like a virus. But we can now use that system to cut and change our own genomes. It's basically a DNA cutting enzyme that doesn't cut randomly. You can give it a barcode in the form of what's called the RNA molecule, where that enzyme will cut in the genome. Let's say you have a terrible gene that's causing heart disease. We take this CRISPR system, we say, here's where you need to go to cut. We can tell the enzyme to go and cut it, put it into your cells, it'll go cut it and destroy that enzyme and delete it. And you can also use it to cut the genome and insert new pieces. So you can both subtract and add DNA at will now, not just randomly. You can tell it where to go, and that's the big breakthrough.
Currently, gene editing is already being utilized in various research fields, including human health, agriculture, and environmental protection. However, when it comes to CRISPR as a tool for human genetic engineering, it is still in its early stages of development. As CRISPR continues to advance as a genetic engineering tool, we can anticipate further scientific breakthroughs. Let's examine the potential advantages and disadvantages of this technology. Particularly in the context of diseases. CRISPR technology holds great potential advantages in addressing diseases. It offers the promise of more efficient and targeted treatments. However, caution must be exercised when applying it to humans, animals, and plants, as it may inadvertently promote or accelerate negative environmental phenomena. Furthermore, many potential side effects of CRISPR have yet to be thoroughly analyzed. Is it conceivable that, in the future, we could enhance the average IQ of the population? According to UCLA neurobiologists, research conducted on mice suggests that the answer may be yes. Occasionally, individuals or animals are born with mutations in their genomes. These mutations can vary in size and impact, and many are random and purposeless, having no effect on intelligence. This is evidenced by the fact that gene mutations that occurred millions of years ago still exist in the genes of living organisms on Earth. Over time, evolution has had ample opportunity to correct and eliminate them, yet many still persist. In fact, numerous mutations found in human DNA are harmful, contributing to detrimental effects on everyday life. These effects range from increased vulnerability to certain diseases to mental retardation to immediate death after birth. Looking ahead, CRISPR technology has the potential to enable efficient and affordable treatments for diseases such as diabetes, Alzheimer's, cystic fibrosis, muscular dystrophy, and even rheumatoid arthritis.
We believe we understand how to slow aging. There are genes that predispose you to a long life. We could make offspring, a family that would potentially live a lot longer. In fact, there are drugs that are in development to actually correct genetic diseases, such as vision loss. What we're showing is, in mice at least, we can reverse the age of the retina and restore the vision of old mice. So we'd reprogram the eye to be young again. So we now know that there's a set of genes called reprogramming factors, also known as Yamanaka factors, that are named after this Japanese fellow who won the Nobel Prize in 2012. These factors are used all over the world, even to reprogram skin cells, other cells, to be what we call pluripotent stem cells. These are cells that can be used to make new organs or new blood cells. But what people hadn't tried until recently was, can you do this in a living animal? Or are you just going to mess it up? And what we found out is that if you do it the wrong way, you mess up the animal and it'll die. But what we've shown for the first time in this paper is you can do it in a safe way. And not only that, reverse the clock, make the cells young, and restore how they work, and get back vision.
CRISPR was initially developed as a technique for editing the DNA of living organisms, but its applications and capabilities have expanded significantly. It is now widely used in biomedical research, including studies on human genetics. One of the primary concerns surrounding technologies like CRISPR-Cas9 is accuracy and safety, as mistakes in genome editing can have significant and potentially dangerous consequences for individuals. Despite these challenges, CRISPR technology holds immense potential for various benefits. While it is not an entirely novel concept, it can aid in treating diseases and addressing genetic anomalies. Some scientists have made promising progress, suggesting that CRISPR-Cas9 may be safer than viral therapies commonly used. However, it is important to note that concerns and controversies surround CRISPR technologies due to ethical considerations and questions of fairness. There is apprehension that these advancements might only be accessible to the wealthy, leading to the creation of designer babies with heightened levels of intelligence. Intelligence, however, is a highly intricate trait that cannot be easily engineered. Many researchers acknowledge that intelligence is influenced by a combination of genetic factors and the environment. Genes play a role in the development of the baby's brain, while environmental factors contribute to how the brain is utilized. Consequently, it is conceivable that genetically engineered babies could potentially exhibit enhanced intelligence due to factors such as increased oxygen supply to the developing brain, as indicated by research experiments conducted on mice. The question remains whether achieving similar outcomes with human beings will be possible.
And in the mice, to restore the vision, we have this 3-gene combination of these Yamanaka reprogramming genes. We put them into the eye, and then we turn them on with a drug called doxycycline. It's the same drug we can feed to the mice, turns on the reprogramming genes for a few weeks, restores their vision back to a young mouse, and then we just take away the doxycycline, an antibiotic, and the mice have their vision back. That's the future, that you'll get a delivery of this virus, you'll take the antibiotic for a few weeks, be fully rejuvenated, and the doctor says, come back in a couple of decades, we'll fix you again, or we'll give you some antibiotic in a couple of decades. But then it gets really weird if you engineer your children to have this system, and you have people engineered to be able to be reversed in their age, or let's say they have an accident and their optic nerve gets damaged, or they lose their hearing. Give them a dose, an IV of antibiotics, and they come just like an embryo. They can rejuvenate, they can regrow their optic nerve, regrow their spine, fixed, back like new.
In the future, It is conceivable that our genealogical records could be incorporated into computer databases, and future computer programs might utilize this information. The advancements in artificial intelligence and CRISPR technology may eventually enable the creation of a new generation of human-machine hybrids. However, the safety of such techniques in relation to humans remains an ongoing question. Our understanding of all the mechanisms involved in editing the human genome is still incomplete, and the long-term and short-term effects of gene editing interventions in humans are not yet fully understood. CRISPR has propelled the scientific community forward and holds the potential to enhance our comprehension of the genome. This, in turn, is expected to drive advancements in both artificial intelligence and predictive medicine. The ability to modify gene sequence and correct previously irreparable errors opens up numerous new avenues for research. However, at present, genetic editing technology is still unable to achieve the extensive modifications that would be necessary to create a superintelligent human, if such a feat were ever possible. The corresponding ethical issues surrounding these capabilities deserve significant attention before they become a reality. Thanks for watching. Did you like this video? Then show your support by subscribing and ringing the bell to never miss videos like this.
If you have more time which you want to spend for more knowledge presented in an amazing way, click here before the video ends.